So today we will have class with Dr. Maryann Martone on the fair data and data sharing in open science.
Okay, great. And Koei, let me know. I think that this should be roughly about 60 minutes, but let me know because, you know, I do tend to go off on tangents sometimes. So yes, as Koei indicated, I'm going to talk about the concept of open science and the fair data principles. And I am hoping that by the end of this presentation, you will understand what open science is and the motivations for data sharing or open sharing of data. You will understand why we're doing this. You'll understand what the fair data principles are at a deeper level. So many of you may have heard the term fair before because it's, of course, one of the driving motivations of how we're dealing with data these days. But we're going to go into them a little bit deeper. And then I'm going to also introduce you to the briefly to the ecosystem of biomedical data repositories, which, as I hope by the end of this you will agree, are the integral infrastructure for achieving FAIR. And again, by the end, I hope you understand why it is that we're going into more detail about things that could be seen as fairly arcane. Again, I think I've mentioned, and I'm sure as you've been giving Zoom presentations, it's very hard to see what people are putting in the chat. But if there's a question, Koei, you know, feel free to interrupt me if you think it is relevant at that time. Otherwise, we can deal with them at the end. But I really would like this to be a bit of a back and forth. Okay, so first we're going to introduce open science as really a motivation for a lot of what is going to be presented after this. And this is just a definition from Wikipedia, but it is a movement to make scientific research. And I want to really emphasize that this is all the products of scientific research, not just the article, though that is an important driver of it, but data, samples, software, code, workflows, all of those things, scientific reviews, available openly, and that this dissemination is accessible to all levels of an inquiring society. That's a fairly a high bar to leap over. But the idea that there are many people outside of the scientific workforce that have a vested interest in the types of things that science is producing. And whereas this might have been a purely theoretical concept a couple of years ago to many of you, I think COVID has really, you know, demonstrated, right, that there is a thirst for scientific information. Also, it demonstrated how difficult it is for people outside the academy to be able to interpret the way science works. So open science is transparent and accessible knowledge that is shared and developed through collaborative networks. So it is very much associated with team science, because instead of taking the things you have and holding them close, you freely sort of share them. And of course, there's different you know, degrees of that, and we'll talk about that. It encompasses practices such as publishing open research in open access journals, campaigning for open access, encouraging science to practice open notebook science, and generally making it easier to publish and communicate scientific knowledge. So as I mentioned just a minute ago, there are levels of openness, and we are not at this point advocating that people put their lab notebooks up, but a lot of people do advocate that. So, right, open science, as is anything, is got subtleties and nuances that are sometimes difficult to appreciate. So what is the motivation for this? Oh, yeah, here it is, motivations for open science, right? It's really a consequence of the digital revolution, which is why we're talking about open science. That is, before, when everything was published in a physical form, that physical form had to exist someplace, and not every place had it. Libraries, of course, were instantiated really to be able to keep those physical forms so that people who had access to the library were able to access that. But with the digital revolution, of course, and the internet, it's now possible to distribute things around the globe almost instantaneously. And that is not something that we had before, but this has certainly been one of the drivers for people to open these things up, because before you didn't need a subscription to access an online journal if you were near a library that had that journal. But with online, right, you can see a lot of these things, but you can't get them. But also computation is important, and we're going to talk a lot about that in FAIR. Now it's not just human beings and reference librarians who are telling you where things are. You have search engines and you have algorithms and things that can compute on this information. And these algorithms, especially in the age of artificial intelligence and machine learning, are hungry and they want data. They want access to materials online that they can digest and compute. So these are one of the reasons why NIH is, as we'll talk about in a minute when we talk about the new data sharing policy, really starting to think about how can we make more of these things available freely for people to use in whatever context they wish to use them, right? This idea of there are many motivated people out there who might want access to this information and we do not necessarily know who they are. The main arguments that were used early on in biomedicine, and this has been going on for about 20 or 30 years, I would say, since the early days of the internet, is I would say two main lines of argument. One is that research is largely funded by the public and the public should have access to the results. So I often give a personal example to this, you may have them yourselves, but when one of my parents was very ill, and we had to make decisions about what to do for her care, I kept sending my brothers review articles from some of the major scientific journals on this particular topic. And they kept coming back saying, how come they are asking me to pay $40 so that I can read this article? So being inside an academic institution, you often don't realize how inaccessible a lot of what we have is. And this was one of the motivations for PubMed Central, which is an open access, NIH's open access library of scientific articles, but there's still a lot more to be done on this. And I think, again, a lot of stories, a lot of people came around to open science when they personally experienced how difficult it was for people to get a hold of articles if they were not associated with a major research university. The second one, as I alluded to a little bit already, is that open science equals better science, right? That it's more transparent, that there are more eyes on results so people can look at them and discredit them faster, or credit them faster, right? Science is supposed to be self-correcting, but there's often a lag when the data aren't available, for example. And then again, we talked about this already, increasing computational capacity that can mine biomedical literature and data for new insights in ways that individual humans simply can't, right? There are currently about 30 million articles in PubMed. There's no human that can read them, but algorithms can read them and digest them and use them. Same with very, very large data sets. So we really want to be able to work with data on a scale that really is out of reach of your individual researcher, either to produce it or also to analyze it. So these are some of the major motivations for why there's a move towards open science. And I think COVID is a really good example again. And one can argue, I usually start off this type of lecture with trying to convince scientists why they need to be able to, why they should be publishing in open access and why they should put data out in open archives. And I think COVID made that argument better than I could. And that is, in times of health emergencies, global health emergencies like COVID, we have to scrap pretty much all of our practices, right? Journals said, oh, okay, anything that's related to COVID, I'm going to publish in open access. There were major perspectives in some of the main journals saying, what we really, really do need to get this information out there more quickly. And we want to be able to see the data, see the underlying a code, for example, that was used to analyze this. And why was that? Well, because in fact, we saw immediately what the impact was of not having this information. So early on in the pandemic, there's still fights about hydroxychloroquine and whether it works or not, and I'm not going to get into that. But Lancet published a major paper that said, no, it doesn't work, that halted global trials for hydroxychloroquine for COVID-19 because they said, oh, there's fears of increased deaths, which is what it showed. But a lot of people had questions about it and they couldn't get a hold of the data. And because they couldn't get a hold of the data and they had real questions about how this worked, Lancet ended up retracting that paper and not making it available. And yet the fact that this was put out there and people couldn't get the data, so they couldn't look at it for themselves, meant that they halted trials. And so that directly translated on a huge impact into people's lives. And I think we have to remember for open science, right? it's always an emergency for someone. It's not necessarily a global health emergency, but if you have cancer, or you have a lot of type 1 diabetes, or any of these things, right? There's an urgency to what it is that science you want science to do for you. So I think COVID again has really, really put the spotlight on open science. Okay, so I'm really going to focus the rest of the lecture on open data. As one of the things that we want people to be able to share. That COVID example gives, I think, makes that very, very clear. It's not just what you say about the data that we want out there. We want the data sets themselves. And that COVID example, I think, gives you a really good feeling of why the data themselves are important, right? Because scientific papers are interpretations, and they will make conclusions based on a data set, but if somebody else reanalyzed that data set, they might come to different conclusions, right? We see that a lot in the sort of arguments about reproducibility. So just a few terms here. I'm going to talk mostly today about what we call long tail data, because this is really tailored to you and your careers. And long tail data is a term that is often used to refer to the types of data sets that would be acquired by individual researchers in the course of their research. And they may collaborate with a few people, but they're generally smaller data sets on smaller numbers of subjects. They're often got very complex experimental paradigms that lead to these particular types of data sets. And of course, the results of these are published in a paper, and there's claims that are made about that data, okay? And I want to distinguish that from so-called big science. Big science are these prospective studies. NIH and a lot of funders will fund where there are thousands and thousands of subjects. There are common protocols across all the sites that are gathering data, and they tend to come together into a dedicated database, which makes this data available, okay? So as Neil McKenna, who talked to you yesterday, often says, long tail data or small data done right are big data, okay? But I'm really going to focus on how you should treat the data that you acquire in the course of your studies to make it what's called a primary product of research. And what do I mean by this? I think right now, even though you're very early in your careers, you know that the scientific paper in a peer-reviewed journal is the currency of science. That is what you are judged by. How many papers have you published, and what journals have you published them, and who is using those. So all of the metrics we have, like H-index and other types of journal impact factors, are based on how many other people cite them. So the idea of treating data sets as a primary product of research means that you will publish your data the same way you would publish an article, and it would be subject to the same types of tracking that we have for articles. So who made this data set, how many people have used it, right? What conclusions have been drawn from it. So when I say a primary product of research, that's what I mean. It sits alongside the paper and not underneath the paper. And why do we want to do this? Again, already went through the arguments of open science, transparency, like you saw in the COVID example. You want people to be able to reuse that data. Maybe they have a need for that particular data set, and you've studied something that they were thinking about studying, and they can then say, oh, I can go and use this as a comparison, or maybe just base their study on your data. And then, as I just mentioned, aggregation to be able to increase the power of those data sets, being able to take data sets that are on similar things, combine them together, and then use the power of artificial intelligence and machine learning to be able to look for correlations amongst these data sets that were produced independently to derive new insights. And I'm not going to go into this today, but I have some very good examples if you're interested from the spinal cord injury community, where they are really using this approach to come up with new and novel and powerful hypotheses that seem to translate better from the individual laboratory to the clinic. So that's a talk for another day, but this is the reason why we're doing this. Okay, so I usually actually, again, have a whole section in here about the power of open data. That spinal cord injury example I showed you, and some of the other ones that I mentioned, and some of the other ones that I used. Because again, I'm mostly trying to convince scientists that they should view their data this way. However, this past year, NIH released a new policy on data sharing, which sort of takes the "why should I" argument away, because NIH is now saying, and you will, okay? So NIH's new data policy, which is going to come into effect on January 25th, 2023, all grants submitted after this time are going to have to pay attention to this new data policy from NIH. And it basically says, all data must be managed, and we'll talk about what that means in a minute. Most data should be shared, okay? So they are now issuing a blanket policy that says the expectation is that you will be sharing your data as we'll get into it in the subsequent slides, in a very specific way when you produce it as part of an NIH-funded study. And the reason why it's most data should be shared is we understand, and even the open science proponents understand, that not all biomedical data can be shared. We are dealing with sensitive data, we're dealing with patient data. We know that we cannot share individual subject information on human beings on the web. So there is a little phrase that is used by the open science community, at least in biomedicine, as open as possible, as closed as necessary. But there's a shift, a very subtle shift, which you might appreciate here, is that in the past, it was just assumed that everything was going to be closed and you would dribble out as little bit as possible in certain circumstances. Now I would say the expectation is that everything will be shared and you need to justify why you can't share it. So it's essentially a bit of a flip, okay? So NIH is now going to require that you submit a data management and sharing plan. How are you going to manage your data and how are you going to share it? It needs to be approved by NIH, and you are expected to actually adhere to that when you're doing your research. So Jeff is going to talk to you tomorrow about the data management part of it, but I am going to give it's impossible to talk about data sharing without talking about data management. So I'm going to go a little bit into this here. So this is one of my favorite videos. Sometimes it's problematic showing these videos on the web. So I'm hoping, or via Zoom, so I hope everybody can see it. It's a 4-minute video, but I think it really illustrates extremely well why it is that NIH is having this policy and why it is that data management and data sharing are considered to be two sides of the same coin. So I'm just going to play it. I beg your patience, and Koei, let me know if it's not coming through. This was done in 2012. It's a little long, but I think you will enjoy it.
Maryann, the sound need to turn on.
What? You can't hear the sound?
Yes, video sound.
Oh,
I cannot hear.
I can hear it extremely well. Yeah, I don't know how to get these things to go. I think it played just fine last year. Let me see if I can figure out what the setting is. Video. No, I can't.
Maybe make sure you shared your sound with us.
Okay, I'm looking at my.
I think you are using an earphone and mic.
Oh, that's it. Earphones. Okay, let me take that off. Let's try it again. Can you hear me still? Oops.
Yes.
Okay. I tell you. All right, let's try this.
Hello, my name is Dr. Judy Benign. I'm an oncologist at NYU School of Medicine.
Is that working?
Hello, Dr. Judy Benign.
I read your article on B-cell function. I think that I could use the data for my work on pancreatic cancer.
I am not an oncologist.
I know, but I think I could use the data for my work on pancreatic cancer. Do you have the data?
Everything you need to know is in the article.
No. What I need is the data. Will you share your data?
I am not sure that will be possible.
But your work is in PubMed Central and was funded by NIH.
That is true.
And it was published in Science, which requires that you share your data.
I did publish in Science.
Then I am requesting your data. Can I have a copy of your data?
I am not sure where my data is.
But surely you saved your data.
I did. I saved it on a USB drive.
Where is the USB drive?
It is in a box. It is in a box at home. I just moved.
But can I use your data?
There are many boxes. So many boxes. I forgot to label the boxes.
Hello again. Thank you for sending me a copy of your data on a USB drive. I received the envelope yesterday.
You are welcome, but I will need that back when you are finished. That is my only copy.
I did have a question.
What is your question? You might find the answer in my article.
No. I received the data, but when I opened it up, it was in hexadecimal.
Yes, that is right.
I cannot read hexadecimal.
You asked for my data and I gave it to you. I have done what you asked.
But is there a way to read the hexadecimal?
You will need the program that created the hexadecimal file.
Yes, I will. What is the name of the program?
Citation.
I do not know this program.
It was a very good program. The company that made the program went bankrupt in 2007.
Do you have a copy of the program?
I do not use this program anymore because the company that made it went bankrupt. Maybe you can buy a copy on eBay.
Act 3.
I have good news.
You again.
I talked to my colleague. She knew a person with a copy of the software.
Then why do you need me? Everything you need to know about the data is in the article.
I opened the data and I could not understand it.
If you have the program, you will find it is clear.
Well, I noticed that you called your data fields SAM. Is that an abbreviation?
Yes, it is an abbreviation of my co-author's name. His name is Samuel Lee. We call him Sam.
I see. And what is the content of the field called SAM1?
Ah, yes. SAM1 is the level of CXCR4 expression.
And what is the content of the field called SAM2?
That is logical if you think about it.
What is the content of the field called SAM2?
I don't remember.
What about SAM3? Is there a guide to the data anywhere?
Yes, of course. It is the article that is published in Science.
The article does not tell me what the field names mean. Is there any record of what these field names mean?
Yes. My co-author knows what the content of SAM2 is and SAM3 and SAM4.
Can I talk to your co-author?
I'm not sure.
I would very much like to talk to your co-author.
Well, he was a graduate student. He went back to China 2 years ago.
Can I have his contact information?
He is in China. His name is Sam Lee.
I think I cannot use your data.
You could check the article to see if what you need is there.
Please stop talking now.
Okay, so I think you can appreciate from this why now data management and data sharing are tied together. And we're going to have an example where I'm going to ask you to critique a data set in this, using some of this information to understand why it is that we have to focus on both of these if we want to make effective use of data. And this particular scenario is extremely common. It is extremely common even today, even if you don't keep your data on USB drives in boxes, even if you have it stored on local computers, if you have it on various places, all of the things that you see happening here happen today. So, really what NIH is asking for and what is good lab practice is that instead of of employing these ad hoc data management practices where you yourself, or Sam Lee in this case, understood how he collected his data and how he organized his data and what his data meant, but it's really specific to that individual. The PI clearly had some procedures in place, which says, well, you got to put it on a USB and you need to give it to me and I will put it in a box. But we can contrast that sort of ad hoc data management for what would be considered good practices that are optimized for reuse. So for example, here, we say that when one's planning one's project, I've created plans for managing my data that are designed to streamline its future use by myself, my lab, and others, right? I organize my data so that others can navigate, understand it, and use it without me being present. So you don't have to hunt down Sam Lee and find out, where he is right now, may have gone into industry, may have decided that he didn't want to be in science. You are able to hunt them down. You are able to understand it without having to do that. And I save my data in a manner and location designed to maximize opportunities for reuse by myself and others. So when we say that you're managing your data to be able to share it, this is what we mean. And I want to say, because you already saw that in some of the boxes there, when one is managing one's data properly, the one who is most likely to benefit from that, first of all, is you, because you can find your data, you know what it means. If somebody has a question about your data, a journal comes back and says, oh, I need the data underlying a figure, you've managed it properly so that you are much more likely to be able to use your own data in the future. You're able to find it. So you and future you are one of the major beneficiaries of good data management. Yes, we emphasize here reuse by people that you don't know, right? That's the open science part of it. But it is also more likely that the ones who are going to benefit the most from this are not just you and future you, but also your principal investigator, also your colleagues, if you want to collaborate around data, right? You now have practices in place that make it easy for you to get this data, understand this data, and just send it. And as we'll talk about when we talk about data repositories, ideally, just like you send a link to a PubMed Central article right now, you will just send a link to an online data repository where you can say, here, everything you need to know is there. And if you have a question, please ask me, okay? So instead of just pointing to the paper, we say, go here, you can find it, and I have organized it and published it in a way that makes it understandable to you. So the question then might be, well, how do I do that? Okay, how do I do that? And in the new NIH data sharing policy, there's two things that really quite stand out. There's a lot of details in there, and I encourage you to read it. In fact, I should have done that in anticipation of this lecture. I'll do that in the future. NIH encourages data management and data sharing practices consistent with the FAIR data principles. And the NIH strongly encourages the use of established repositories to the extent possible for preserving and sharing scientific data. Okay, so NIH has tied this policy to these things called the FAIR data principles. And as I've mentioned before, data repositories are really at the front lines of FAIR. So I'm hoping that by the end of the next section, you will appreciate why FAIR is powerful, what it means, and also why it is that we are looking to scientific data repositories to help us achieve FAIR. Okay, so this next part is really going to be on the FAIR data principles for scientific data management and stewardship. This was an article that was published by Mark Wilkinson and many, many others, including Jeff and myself, back in 2016. Because of projects like dkNET and also the Neuroscience Information Framework, which is another project we ran, we knew firsthand what the difficulties were in trying to deal with data that was out on the web. We knew what the bad data management practices were. We knew the lack of documentation. We knew everything else. And so Maryann Mons came up with this very succinct acronym, which basically said, if one wants to reuse data, and I do want to say right here that FAIR itself is agnostic to open, right? It doesn't really care whether anyone can get this data because it's freely available, or whether it's behind a firewall because it's human subject data. It's any data that's out there on the web. He said, if you really want to be able to reuse this data, then you need to make it findable, accessible, interoperable, and obviously, you need to be able to reuse it. So a lot of people know this high-level acronym now. It's really kind of made its way into the scientific consciousness. I wouldn't say at the level of bench scientists necessarily, though, but certainly at the level of of those who are tasked with building data repositories and other things. There's still a lot of work to go. But by and large, people now understand that data should be fair. What they don't necessarily know is, for some reason, it just likes to skip my slides. Okay. What they don't necessarily know is what the details are about fair. So before we go into those a little bit, I want to give a couple of definitions, right? One of them is, you'll notice that the fair data principles say that they want to make things findable, accessible, interoperable, and reusable for both humans and machines. That is, you want a human to be able to do all of these things, but you also want computers to be able to assist you with this and do a lot of things without you having to intervene. So I want to introduce a concept here, which is machine readable, machine actionable. There's many different ways that one can sort of state this. But the idea is that data, and there's always a little meta in front of it because we're talking about both metadata and data, and I will explain what metadata are in a little bit more detail in just a moment, is published in a format that can be easily processed by a computer without human intervention while ensuring that no semantic meaning is lost, okay? So it basically means that you are preparing your data in a way that a machine or a computer, I should say, can interpret that data and it knows what it is allowed to do with that data, okay? So it is very intimately tied to having some sort of structure to your data and text, or embedding special tags inside of your data that a machine can process. And what I mean by this is, I think, illustrated in my next slide, which is CSV file, or isn't it? So CSV stands for comma separated value. You would know this by its most common name, which is a spreadsheet. This is a Google spreadsheet. And so you have columns, and then you have values inside of those. You have, and that what this really means is that underlying this, there's a comma, and the computer knows how to interpret it. So here's a CSV file, common format, same as Excel, same as Google Sheets. And we actually saw this several times when we were looking at supplemental data as part of dkNET and the NIF project. So these on the surface both look like CSV files, right? They're tables. You might see something like this in a paper. But in fact, they are really quite different in terms of machine readability. The top one, oops, the top one is a picture of a table. So this is actually an image. If I look at its data type, it's an image. And if I select it, you see I can cut it out and I can put it in. But it's not actionable by a computer. The second one is an actual spreadsheet. So it is truly a CSV file. And here I can select individual values. I have something called age, and that age is a number, and the computer knows that this is a variable called age, and an age is a number, and therefore I can compute with it, right? I can say this one is older than this one, okay? So this is an example of actionable data. This is an example of the type of thing that the little bear was talking about in his particular case, where he says, oh, it's all in the paper. But most of the time, that's a picture of a table. And trying to extract that information from papers is fraught with difficulties. So this is what we mean by actionable data. I'll also use the word machine readable, machine processable, right? A computer can look at this, and because they can interpret it, there are operations that they know that the computer can perform on it. This is very integral to FAIR, which is why I wanted to make sure that I introduced it before we started talking about FAIR. Okay. The second concept that you need to know is metadata. And metadata, very simply, is data about data. Apparently it's so important I said it twice, data about data. And there's a couple of types of metadata. It's a very, as all of the concepts I'm presenting at a very, very high level, and there are little subtleties that I don't think I need to tell you about. The simplest type of metadata is about the file. So that file that I showed you before is a CSV file, and maybe it's a few kilobytes, maybe it's a few terabytes, right? So it tells you about that data file. The data I'm mostly going to be talking about here, the metadata I'm mostly going to be talking about here, is something called descriptive metadata. Descriptive metadata describes a data set for purposes such as discovery and identification. It can include things like abstract, authors, what conditions you studied, right? It basically is providing context for an individual data set. So when we're talking about metadata from here on, this is the type of metadata that I'm going to be referring to. So now let's take a closer look at FAIR. And I'd like to use this little slide as an introduction to it, because a lot of people who were instrumental in getting FAIR put together kind of sniff a little bit. They're like, well, people use the word FAIR, but they really don't know what it means. And I like to say that FAIR is like mom and apple pie, as we say, saying in the United States. Who would not want it? Like, who would be against that? So of course, everybody wants their data to be fair, and everybody's going around saying, yes, my data is fair. But in fact, FAIR actually, if one reads the paper, which I hope you all did, it tells us about 15 things that encompass what FAIR requires, if you will, in order to be achieved. So it has all of these things, which I'm sure were not at all understandable by a majority of you, and that's perfectly fine. We're going to go through some of them, and some of them really don't apply to you. But they say things like data should be assigned a globally unique and persistent identifier. Data are described with rich metadata. Data have a license associated with them. Data use vocabularies that follow FAIR principles. So FAIR gives you a set of guiding principles, if you will, and recommendations for how you are to make your data findable, accessible, interoperable, and reusable. So in the next part, we're going to go through some of these, not all of them, don't worry, and try to understand what they mean. So I'm going to spend most of my time on the findable one, because as the FAIR principles purveyors say, if you can't find data, then all the rest of it is meaningless. You saw that in that little video, right? If you can't get a hold of the data, then there's no point in having a discussion further. So the first two are listed in order of importance. Number one is that data are assigned a globally unique and persistent identifier, what is called a PID, a persistent identifier, and that data are described with rich metadata. So your first response to that, given that video, might be, why is a globally persistent and unique identifier the most important thing about FAIR? And as again, people who interpret FAIR, the ones who were instrumental in getting it out there, say principle F1 is arguably the most important because it will be hard to achieve other aspects of FAIR without globally unique and persistent identifiers. Hence, compliance with F1 will already take you a long way towards publishing FAIR data. So let's spend a little time on these PIDs. PIDs are basically unique in the world, and by the world, we mean the digital world. And they are stable, that is, they persist. So unique means they identify only a single object, okay? And persistent is they identify that single object for all time, so they're never reused. I'm sure all of you have experienced broken links on the web. And sometimes those links just say, I'm sorry, I'm broken. But sometimes links actually go to something else. Right? It's a domain name that got lost by an individual and it was taken over by somebody else. And so you go to this thinking that you're going to get to a scientific site, and now all of a sudden it's some sort of spam site, right? Where they're asking you to buy hair products, right? So that is a unique identifier because it's the only one in its existence, but it's not persistent because it now can point to multiple things. So both of these things are important for FAIR. The other thing that's really important is that they're really only persistent and unique because organizations stand behind them. So organizations like the Internet Registry say, well, you can only have one domain that has this particular address, but we don't care who has that domain, right? If you stop paying for it, somebody else can have it. So there are organizations, data repositories, as it turns out, that are responsible for issuing these things for biomedicine. And it's those organizations that make them fair. It's not the fact that there's an identifier itself. The organization has to make sure that that is never reused and that it only points to one object, okay? And we're going to see that one of the reasons that this is so important and why there's that 1-2 punch of F1 and F2 is that these persistent identifiers allow a digital object like a data set to be reliably tied to their metadata. And that's really what's so important about them. So here are some examples of persistent identifier. You've encountered them all the time if you deal with biomedicine at all. First of all, in articles, there's something called the digital object identifier. Digital object identifiers were actually developed by the publishing industry because when they first put all their content online, everybody would build up a site and then they, a publisher would have a journal and then it would change its link structure or it would move to a different domain, it would merge with another company and everything broke. So this DOI was invented in order to point to an article, and it doesn't matter underneath where you're storing it, doesn't matter what the underlying website is, it will always point to this particular article. That same concept is the ORCID. So we asked you all to sign up for ORCIDs. That ORCID is the thing that uniquely identifies you. So if there are 100,000 Sam Lees, there's only one Sam Lee who published that data set, and that ORCID is ascribed to him, okay? And you've also seen the research resource identifier, which is the globally unique identifier for research resources that Anita talked about the other day. So there's many, many types of PIDs. They are managed and maintained by individual organizations that ensure that they are unique and persistent, and they are the ones responsible for assigning them, okay? They are the ones who are responsible for assigning them. So again, why is it that we have them? Well, we already talked about one of the main reasons for the DOI, which was that they wanted to stop all this link rot, all of these broken links, which happens all of the time, right? And we're also going to talk about the fact that, we're also going to talk about how important they are, as I mentioned, for tying an object to its metadata. But again, always remember that these are social contracts and it is not something that you yourself can do. You have to go to a registry to have one of these things assigned for you, okay? So this is lesson 1 from this. You cannot assign PIDs to yourself or your data. You need to go to specific services that do this for you. And as we will talk about, that entity in this case is going to a data repository that provides these persistent identifiers. Just like you don't assign an identifier to your article when you publish it, the publisher does that. That is a function of data repositories. So now let's talk about metadata, right? Data are described with rich metadata is number 2. And again, in terms of ranked importance, that's important. So what exactly are rich metadata? You will notice that in both the Fair Data Principles and also the NIH data repository, there's a lot of, the policy, there's a lot of wiggle room. And there's a lot of wiggle room because biomedicine is so vast with so many different domains, with so much, so many different types of data and paradigms that they're dealing with, that issuing blanket statements about what it is you must do in terms of a metadata model, for example, would quickly lead to people just abandoning these things, right? They're like, I'm sorry, this does not apply to me at all. So there's a lot of latitude that is given to individual communities to be able to interpret this themselves. And in the case of rich metadata, there is no little document that says, these are rich metadata for biomedicine. But I like this statement, right, that was issued by a Supreme Court justice, very famous statement. It was actually issued to talk about pornography, but it says, I shall not today attempt further to define the kinds of material I understand to be embraced within that shorthand description, and perhaps I never could succeed in intelligibly doing so, but I know it when I see it. And I would say right now that when one encounters rich metadata, you know it when you see it, okay? Even if a particular data set doesn't have this particular attribute. So let's give some examples of that. This is a data set that is actually deposited in an archive called DANDY for electrophysiology, mostly neuroscience. And when you go to their landing page. So you have an identifier, the identifier takes you to that landing page. This is what you encounter. You see that the title of the data set is 202.006.11 AIBS mouse, and that the description is the Allen Institute June 2020 mouse patch-seq IceViz data. So I would consider this an example of unrich metadata. I look at this title and I don't know what it means. I wouldn't even know how to search for it because it doesn't tell me anything. I might be able to put in patch-seq and mouse and get something from the description, but it really doesn't tell me very much about it, okay? We would never publish a paper with a title or a description like this, right? We have a detailed abstract, we have a detailed title. So one of the things I hope to impart to you here is that if you start to think about publishing a data set like you would publish a paper, you know that there are certain things that you should provide. You would provide the authors, the affiliations, you would put an abstract. There's other structure that you would put in there, right? We know how to do that. But at least this part here, which is very, very much applicable to data sets, more often than not, what you will see in public repositories is what you saw for that data set. Virtually nothing there. Let's contrast this to a data repository that my team, of course, is curating, which is why we think it's, of course, particularly good. But this is something called the SPARC project, Simulating Peripheral Activity to Relieve Conditions. And here, submitters are required to provide us with this data. And we have human curators who make sure that they do, because without the human curators, all of SPARC data sets would look pretty much like what you just saw. So here you'll notice there's so much metadata we couldn't fit it in a single screen. But when you go to a page describing this data set, when that ID takes you here, you see a meaningful title with meaningful information in it. You see a full author list. It tells you how to cite this data set. And if we have time, we'll talk a little bit about that later. It has a data description. It provides a link to an experimental protocol, so you know how that data was acquired. It gives you notes that you need to know in order to use it. It provides information on the subjects and samples. It actually provides you link to code that can be used with this data. It gives you keywords. It gives you the originating publication where this data set was first described, so you can get that additional information. So if one was to encounter these two data sets, which one would you be more likely to reuse? If I found this through a web search, if I accessed them even by a paper, if I shared these with my colleagues, if you tried to use these data a year after you produced them, which is the one that would be more likely to be able to be reused? Obviously, it is the one that has more metadata, okay? Now, maybe the person who did this didn't move back to China, and maybe you can still access it. But in fact, if I were to encounter this as a standalone object, I would be much more likely to reuse the data set on the left. So this is really what we mean by rich metadata, right? So when you have unique identifiers and you have rich metadata associated with them, it is a very powerful combination. So the lesson here is more metadata is better than less metadata. And if you think about publishing data like you would publish a paper, you then start to think, what information do people need to understand it and use it? Because you're no longer talking to yourself with variable SAM1, SAM2, SAM3. You're saying, what would somebody else need to be able to use it? Okay. So let's talk a little bit more about these identifiers, because you see they're kind of throughout the FAIR principles. So number one, oh, now there's vacuuming outside. This is the annoying thing about giving lectures during COVID. So let's just look a little bit at the accessibility requirement. It says, metadata data are retrievable by their identifier using a standardized communications protocol. And the protocol allows for authentication and authorization procedure where necessary. So again, FAIR is not equivalent to open. If you need to put these behind authorized access, you can, but the machine ought to know how the human needs to know what they need to do in order to access it, and the machine needs to be able to interpret that. So there needs to be a way for it to say, yes, you have access to this data set, and therefore you can go and get it. And that's what most online repositories do. So in order to understand that first one, however, which I'm going to spend a few minutes on, you need to understand the difference between structured and unstructured metadata. okay? This is a very important concept in computer science and understanding data. Structured data or structured metadata are data organized according to a data model, right? An abstract model that organizes the elements of data and standardizes how they relate to one another and to properties of real-world entities. Unstructured data or metadata is information that doesn't have a predefined data model or is not organized in a predefined manner. The classic example of unstructured data and metadata is just free text. So when scientists write free text in a paper, you need special algorithms that are increasingly AI-based to try to read that and understand what it is that you're trying to say, okay? And there are intermediates before that. But if we think about structured metadata, what we're really thinking about is a CSV file, for example, a database, something that provides a machine-readable model that the computer can interpret. So here's an example of unstructured data, metadata. Subjects comprised male, n equals 6, and females, n equals 6, C57, black 6 mice, aged 25 days. That would be the type of sentence you would have in the method section, right? If there were a structure, data structure behind it, it might say something like, well, there's something called a subject group, and subject group 1 is comprised of organisms who are, in this case, a mouse. They have an age, which is 25 days, and 25 is an integer, but it's an age, and that the unit is days. Here's its strain, its sex, and there's a number called 6, okay? And then you would have subject group 2. So if you wanted to find out how many subjects are in group 2, you would just issue a query to a database, and it says, well, I know that because there's something called number. So it would say select number of subjects from subject group 2, right? That's how structured data works. So why is there this emphasis about being able to retrieve this data using an identifier? And that's because identifiers can then be used to reliably retrieve structured metadata and use it in another context. One of the best examples of this is reference managers, okay? Because if you look at an article, there's a little database behind that has tags, and it says, right, this is an author name, and this is a title, and this is a publication year, right? So it gives a little context to what you're seeing inside of an article. So I usually like to give just an example of this, of how powerful it is and how it makes your life easier by actually using an online reference manager to take something like this. This is, of course, text, but what you don't see is that embedded underneath that text is a set of tags that allows a computer to interpret the metadata that is associated with this particular article. So I have an online reference manager called PaperPile. This one, of course, is not in PaperPile because we just published it. But if I say add to PaperPile, it's going to go and say, first of all, I know that it's a data type article. And I know for a data type article, I would have a title, I'd have a journal, I'd have authors. So then when I go to my reference manager, okay, and I look at Antibody Watch, it knows exactly about this. It knows that it's a journal article. It knows what the abstract is. It knows what the title is. And it can use this now in different contexts, right? You do not have to in your reference manager, like we had to do when they first started and all articles were paper. We had to type in all of this metadata. We don't type in this metadata anymore. We give it an identifier like a DOI or a PubMed ID, and it goes ahead and it grabs that and it imports it in. So it makes it possible to use it in these different contexts, okay? So that's why, again, we now have identifiers. We have rich metadata and structured metadata, and we can do a lot of things with these. And I know I'm going fast through this, but remember, we will have follow-up exercises. So that brings me to the interoperable principle. And here I'm only going to talk about metadata use vocabularies that follow FAIR principles. What you saw was a brief example there, right, of how having well-structured metadata can allow a system to interoperate with another one, right? So my reference manager can interoperate with another system. Now we're going to talk about how we can get down to the data level and how we can perhaps combine data across data sets. So that's why I want to emphasize this particular FAIR principle. So here's a little exercise. This is, again, a typical scenario. We've got 3 different laboratories who are all studying a rat model of type 1 diabetes. Data set 1 is physiological measurements correlated with gene expression in the kidney. Data set 2 is gene expression in peripheral organs. And data set 3 is gene expression in major organs. So here I've created a little fake spreadsheet that brings these 3 data sets together. Let's say I found them. And I see that in data set 1, I have something called age, organism, gene name, and location. In data set 2, I have age, range, subject, gene, and region. And in data set 3, I have age, animal, exp, and sample. And here are my values. I have an age range up here. I have this name of an organism, looks like a strain. I have a gene name, and I have something called CT kidney, okay? Now, I want you to take a look at this and tell me, what are the problems here that you would encounter if you wanted to combine these data? This is where the idea of FAIR vocabularies come in, okay? FAIR vocabularies basically have the characteristics of FAIR data. That is, they have persistent identifiers, there's metadata associated with that, and they also have relationships to other things. That's a FAIR principle we didn't look into a bit. So in fact, there's something called an ontology called Uberon, which is an ontology of anatomical structures. It's a type of controlled vocabulary, which is very sophisticated. And this is a community ontology. It's available to everybody. And you find out the cortex of the kidney actually has this identifier, right? And you know that this is cortex of the kidney because you look at this. The computer looks at this and says, oh, actually, this is cortex of the kidney. And it turns out that renal kidney is a synonym, okay, that's been stored along with it. So it knows all of the synonyms. So you can call it whatever you want, just like I can call myself whatever I want. And if I use my orchid, the machine knows it's still me. Here, the computer knows that, in fact, those 2 things are the same because they have the same identifiers. So really, what you're seeing is that persistent identifiers are more than just data set IDs and articles, that they can be used throughout, right, that data set to identify things uniquely and unambiguously. And when one publishes them with those data, with those IDs in mind, right, you make your data set much more machine processable. A lot of data repositories offer people the opportunity to put these and associate these identifiers with their data sets, but most researchers do not take advantage of that, right? I think largely because they don't really understand what they're for. And yeah, there's always some troubles using identifiers. But really, the lesson here is never pass up an opportunity to use a PID if you have the opportunity. So here, what's really interesting about this is there are actually identifiers for concepts like organism, gene, and anatomical structure. They come from these community ontologies. But then when we start to look deeper into our data, we see that, in fact, these are all the same animal strain. These are all the same rat strain, but you would never know that by looking at their names, right? These are all the same animal strains. There are official strain nomenclatures, but nobody ever used them because they look like this, and they're very hard for a computer to process. They've got all kinds of special characters in them, and they're hard for humans to write. But if one uses the RRID in your paper, in your database, in your lab notebook, then there's no question about what reagent and tool you used, and both you and the computer can identify it. It turns out these are all the same gene, okay? And if we look at the gene ID that's associated with rats, this is in fact the same gene. The fact that it has different names doesn't bother the computer because it only looks at the ID. And down here, we see that, in fact, cortex of the kidney and renal cortex are the same structure, but this is the identifier for cerebral cortex. So again, the computer has no trouble understanding that these 2 things are different. And in a pinch, if you looked at the identifiers yourself, you'd go, well, those are 2 different identifiers, so they're probably different. So I think you can see how powerful, right, thinking again about your data in this machine processable and fair way is. Because what it allows us to do you've already been introduced to RRIDs and dkNET and how you get them, is it allows us to combine across sources. So we can now aggregate to get data together from these smaller data sets into something that's really powerful. And that's what dkNET's research resource reports are, based on the RRID. Because there are databases that use them, because there are projects out there that are trying to validate these things or have reported problems with them, through the agency of this RRID, we can pull all that information together and take that metadata and combine them. Is all of it consistent? No. But again, when you find inconsistent things in data, it doesn't mean that the data are bad. It means that you have a problem to solve. So that is why these persistent identifiers are so important, and they're important throughout FAIR, because they really allow the machine to be able to do things that otherwise would require a whole lot of human effort to find these things, take that data in, reconcile that metadata, and put it together. Again, what I like to say about these, because a lot of researchers are uncomfortable, they're like, well, I said it's cortex and you said it's cortex, but how do we know your definitions are the same? In fact, you don't, right? But before, you didn't even know that you were both talking about cortex because it looked completely in the database. And as I mentioned, if you bring those two things together and they're not consistent, then we either have a problem with the concept and how it's applied, or we have a problem, right, with, or we have a mistake, okay? So there's plenty of good examples where, depending on the technique you use, you might get a different definition of a structure, but we want to expose those. We don't want to paper over them by not ever being able to put two data sets side by side and say, gee, is the definition of renal cortex consistent, or are there some problems here, okay? So again, it's really quite important to think about these things. They don't solve the problem of trying to interpret data, but they do allow you to bring data together so that they are now potentially interpretable in much more powerful ways than they were before. Okay, I see that we are kind of running out of time. So again, here's your lesson. Never pass up an opportunity to do a PID, use a PID, and use RRIDs and other IDs in your lab notebooks, spreadsheets, data dictionaries, and papers when you have an opportunity. Okay, we're running towards the end, and we're getting into the reusable one. And I'll go through this quickly because I think I've brought up most of the points. One, metadata are richly described with a plurality of accurate and relevant attributes. Metadata are associated with detailed provenance. Metadata meet domain relevant community standards. So we're going to talk a little bit about what these things mean. And I'm actually going to give you the lesson of this section before we go through the actual examples. And that lesson here is, think about where your data is going to end up in the beginning, okay? So what that means is, as we've talked about, right, your data, according to the NIH policy and according to good practice, is going to be submitted to a data repository that is specialized for your data type or your domain. Could be just a general repository, but those repositories are going to have requirements about what standards they want used, right? What metadata that they want acquired, that plurality of attributes. And if you know that before you acquire your data, you can plan for that during your experiment. One of the most common problems we have with empty fields inside of data archives is that the researchers do not have that information at hand, so they can't find it, or they never recorded it in the first place because they didn't know that they needed to. So if one understands how to achieve this reusability, the repository is a good place to start, because it's going to tell you what is expected to publish that data, and therefore you can plan your data acquisition accordingly. Again, Jeff is going to go over that on Friday. So dkNET helps. We provide a list of data repositories that are available to you. I think Jeff, in this particular domain, and Jeff showed you that on Monday when we went over it, and you should all take a look at it, and this will be incorporated in the exercise that I give you about FAIR data. But basically, just like all journals aren't the same, all repositories aren't the same. Some of them are specialized for data type. Some of them are specialized for a domain, like type 1 diabetes, for example, may have a repository. Some of them might take anything, like PLOS ONE, for example. And some of them are run by commercial entities, and some of them are run by researchers, and some of them are run by nonprofits, and some of them are run by your library. So there's a rich array of data repositories that are available to you, okay? Generally, we can divide them into 4 different types. As I mentioned, they're data type specific, they're domain specific, they're generalist, so they take anything, or they may be run by your library. And so you have access to them if you are a researcher at that institution. But they are really your ally in FAIR, right? They're the ones who are going to give you the identifiers. They're going to provide searchability. They're going to provide you standards, persistence, all the things that FAIR require. The bulk of that really falls on the repositories to implement, but they can't implement it if you haven't prepared your data properly in the first place, right? They're only as good as what you put into them. And I think that example between the rich metadata where we forced them to put it in because we had curators versus the uncurated starkly shows the difference between that, okay? So for example, if you want to know, well, are there standards that I need to use for my data? You look at the dkNET listing, it says, yes, if you want to put your data in Array Express or Geo, which is a database for microarray, there's something called the Miami standard for microarray data. And it tells you quite clearly the experimental design, it tells you what data it's going to want, it tells you what metadata it wants, it tells you in many cases good experimental practice. So there are a lot of these standards out there. dkNET tries to make it easy for you to access it. There are also entire databases such as fairsharing.org, which if you want to know if there are standards that govern your domain, you can go and look at them there, okay? So understanding at the beginning what standards you're going to have to comply to at the end saves you a lot of trouble. This is, again, data management plans. I won't go too much into it, but what NIH wants you to do is to think about this right before you start, because then not only can you collect your data in the appropriate manner, but you're also going to be able to allocate enough resources for preparing and sharing your data, because doing this type of work at the very end can be very difficult. NIH actually lets you now put on your grants, oh, I'm going to need somebody to annotate the data, or I'm going to need to give some money to this repository because I got 80 terabytes of data, right? So this helps you also allocate resources and request the types of resources you want. And Jeff will go over that tomorrow, okay? Like 2 or 3 minutes, but I'm not sure that I have time really to go over data citation. So I perhaps won't go over that now, so that at least I think there's a lot of comments there that we might want to talk about. But essentially also this requirement for provenance where you put down the title, you put down the authors, you put down the institutions, you put down the experimental protocol, you put down all of the things that you would need to know in order to understand whether you trust that data set or not, is also leading, right, is also absolutely required for a new system of credit that is being developed around data sets where you can cite them just like you cite papers. So if you publish a data set as a primary research object, you will be able to put a citation to that. In fact, best practice is that you do put a citation to that in your paper so that the people who shared that data get credit for it being reused. Okay, so just to sort of wrap up, there's plenty of other slides, but you can take a look at them yourself. We're at the early stages here of a major transformation in biomedicine. Those of us who've been in this domain have been in this since the internet came out in the 90s. I can tell you war stories of the early days of the internet. Of course, that's alien to you because you've only existed in this world. You've never existed in the other world. And that means a lot of these things are natural to you. But changing the culture of biomedicine is going to take a long time. It is going to be a profound change. And there's very much a desire by NIH and others that this not be seen as purely a burden that you have to adhere to, but that there is opportunity here. There's opportunities for new types of careers in data science. There's opportunities for you yourself to manage your data better so that it is much easier for you to do your work, and you yourselves will be able to mine on your old data in order to get better insights. So I really like to say to everybody here that you're at the beginning, right, of this profound transformation. You play a part in it, both in your practices and also participating in giving recommendations, making the tools better, saying what it is that you need. So you can look at it as a burden, or you can also look at it as an opportunity. And I myself look at it as a really exciting thing to have lived through. And I hope you do too. Okay.